The Oxygen Paradox, Oxidative Stress, and Aging. Arch Biochem Biophys. Author manuscript, available in PMC, 2017 April 1. Published in final edited form as Arch Biochem Biophys. 2016 April 1, 595, 28-32. doi, 10.1016/J. Abbreviation 2015.11.015. PSID, PMC 4838776. NIMSID, NIM739379. PMID, 27095211. Author information, copyright and license information, PMC disclaimer. The publisher's final edited version of this article is available at Arch Biochem Biophys. Abstract. Professor Helmut Cees is being lauded in this special issue of Archives of Biochemistry and Biophysics, on the occasion of his retirement as editor-in-chief. There is no doubt that Helmut has exerted an enormously positive influence on this journal, the fields of biochemistry and biophysics in general, and the areas of free radical and redox biology and medicine in particular. Helmut Cees' many discoveries about peroxide metabolism, glutathione peroxidases, singlet oxygen, carotenoids in general and lycopene in particular, and flavonoids, fill the pages of his more than 600 publications. In addition, he will forever be remembered for coining the term oxidative stress that is so widely used, and sometimes abused, by most of his colleagues. Keywords: oxidative stress, aging, glutathione and glutathione peroxidase, singlet oxygen, peroxynitrite and nitric shida, carotenoids, flavonoids. Introduction. I first met Helmut Cees at the University of California at Berkeley in 1979. I was a graduate student working with Lester Packer, Bruce Ames, Paula Timoraz, George Brooks, and Dan Koshland, and Helmut was a visiting professor from Düsseldorf, Germany. Little did I know, at that time, that Helmut would become a valued and dear friend over the succeeding 35 years. Helmut became a frequent visitor to Berkeley over the next couple of years, but I suspect his enthusiasm was more a romantic interest in his future wife, Nancy, than an inability to stay away from the research we were all doing. In the end, Helmut managed to steal Nancy away from us, which was certainly one of the most intelligent things he has ever done, and that's saying something. Over the years, Helmut continued to collaborate on many projects with Lester Packer, including the early Oxygen Radicals Gordon conferences, and the founding of the Oxygen Club of California. We are celebrating Helmut's many achievements over a long tenure as editor of Archives of Biochemistry and Biophysics. But it should also be remembered that Lester Packer preceded Helmut as ABB editor. As a member of Les's lab, I often dealt with ABB papers in those early days. There are frequently interesting relationships and even apprenticeships in scientific families, and so I suppose you could describe Helmut and I as cousins. I took a term as president of the Oxygen Club of California, OCC, from 2002 to 2004, when Les Packer stepped back to become founder and honorary president. No huge surprise then to recollect that Helmut Cees became OCC president from 2004 to 2008 and, of course, did a fine job with the organization. Helmut and I then reversed terms as presidents of the International Society for Free Radical Research, SFRR International, he from 1998 to 2000, and I from 2003 to 2005. Since both the OCC and SFRR International appear to be thriving, I will take the liberty of assuming that at least neither of us did irrevocable damage. Helmut's contributions to our understanding of hydroperoxide metabolism, glutathione metabolism, peroxidases, and selenium have been tremendous. Similarly, Helmut's research made a huge impression on our appreciation of carotenoids, especially lycopene, flavonoids, flavonols, and polyphenol. Helmut has also made major contributions in the singlet oxygen field, and to our understanding of nitric oxide and peroxynitrite metabolism. The Distinguished Scientist Fellowship Program of King Saud University indicates that Professor Helmut Sees has published over 600 scientific papers and book chapters. He won the Werner Heisenberg Medal, Alexander V. Humboldt Foundation in 1999, an honorary Ph.D. from the Universidad de Buenos Aires in 1996, the ISFE Prize in 1994, the Claudius Galenas Prize in 1990, the Ernst Jung Prize for Medicine in 1988, the Silver Medal of the Karolinska Institute of Stockholm in 1986, and the FEBS Anniversary Prize from the Federation of European Biochemical Societies in 1978. Professor Helmut Sees is a past president of both the European Society for Free Radical Research and the International Society for Free Radical Research, and is a member of the Distinguished Editorial Board of the journal, Free Radical Biology and Medicine. He has chaired both the Oxygen Radicals Gordon Conference and the Oxygen Radicals Gordon Conference. He is a member of the German National Academy of Sciences, and a Fellow of the Royal College of Physicians, London. I could easily fill several pages of this article with Helmut's achievements, but I think that the point is made with even this abbreviated list. Helmut's famous paper on the concept of oxidative stress was extremely influential for many, if not most, of us working in the free radical field, 1-4. The impact of the simple statement that oxidative stress denotes a shift in the pro-oxidant/antioxidant balance in favor of the former really cannot be overestimated. In addition, Helmut presented the whole concept of oxidative stress within the context of normal aerobic life, and not as some rare or aberrant occurrence. Indeed, he proposed that diverse biological processes such as inflammation, carcinogenesis, aging, radiation damage, and photobiological effects appear to involve reactive oxygen species. This field of research provides new perspectives in biochemical pharmacology, toxicology, radiation biochemistry, as well as pathophysiology. Helmut's oxidative stress concept is, of course, a complex outcome of a much more simple phenomenon for which I coined the term, the oxygen paradox. 5. Simply stated, the oxygen paradox proposes that although oxygen is essential for aerobic beings, it is also inherently dangerous to the very same life forms. In other words, it is very difficult to live without oxygen, but it is also very difficult to live with oxygen. Oxidative stress and the oxygen paradox, therefore, form a complementary dyad which leads us to look for the mechanisms by which life on Earth has managed to find ways to profit from an oxygen environment without immediately falling prey to the dangers of oxidation. Glutathione, selenium, and glutathione peroxidases. Having worked with Britton Chance at the Johnson Foundation in Philadelphia in the 1970s, it is surprising that Helmut Sees became a champion of the antioxidant role of glutathione peroxidases. This is because the Chance Lab, in those days, was a staunch proponent of catalase as the major intracellular peroxide metabolizing enzyme, a position strengthened by the rather amazing catalytic activity exhibited by catalase. The turning point, at least for Helmut Sees, seems to have been discussions with Leopold Floh that resulted in a series of challenge experiments that the two scientists then performed. The results of these groundbreaking trials led both to agree on the importance of glutathione peroxidase, 6-8, and, ultimately, changed the direction of an entire field, and even the mind of Brit Chance, 9. Of course, we now know that catalase and glutathione peroxidases are largely compartmentalized, which limits their actual competition in vivo. In the absence of such geographic knowledge, however, the studies of Sees and Floh 6-8 can fairly be described as seminal. Helmut Sees went on to study glutathione metabolism and transport, and to establish that proteins could undergo glutathionylation and glutathionylation both in vitro and in vivo. Years later, when my own group found that glutathionylation can modulate the activity of the proteasome, and that such glutathionylation reactions may actually be used physiologically to regulate proteasomal activity and selectivity during cellular stress, 10, we leaned heavily on Helmut's work as we tried to understand the chemistry and biochemistry of glutathione reactions. Most glutathione peroxidases, but not all, are selenoenzymes, and selenium metabolism and chemistry became an important part of Helmut-Cees' research, 11 to 13. In typical Helmut style, however, he was not content with chemistry and biochemistry alone, but went on to make significant nutritional contributions dealing with selenium bioavailability, intestinal uptake, and incorporation into selenoproteins, 14 to 17. Singlet oxygen, nitric oxide, peroxynitrite, carotenoids, flavonols, and flavonoids. Helmut-Cees has published a wealth of experimental evidence detailing the importance of various carotenoids, particularly lycopene, in protection against singlet oxygen and UV light, and of the importance of flavonoids, flavonols, and carotenoids in human health, 18 to 28. At the same time, he also managed to undertake groundbreaking research with nitric oxide and peroxynitrite, 15, 35, 36. These studies span an immense scientific arena, as they run the gamut from chemistry and biochemistry, to nutrition and intestinal absorption studies, to physiology, pathology, and medicine, 18 to 22. Very few, if any, researchers in the free radical field can claim to have undertaken anything like the comprehensive approach to the study of antioxidants that Helmut-Cees has made a trademark. Oxidative stress. Certainly, no single term is so clearly associated with the name Helmut-Cees as is oxidative stress. For many years, Helmut's concept of oxidative stress guided our investigation of antioxidant enzymes, antioxidant compounds, and especially damage removal and repair systems such as lipolytic enzymes, proteolytic enzymes, proteasome, LAN, autophagy, and DNA repair enzymes, 39 to 43. Although today the emphasis in the field has shifted to more discrete and trendy, redox control of signaling pathways, oxidative stress still appears to be an entirely valid terminology when applied to the frankly toxic effects of very high concentrations of oxidizing species, or repeated or chronic exposures to such agents. Thus, toxicologists, pathologists, and many physicians will likely continue to find value in, and applicability of the term oxidative stress for the foreseeable future. There is no doubt that oxidative stress has been misused by some, as have fluorescent dyes, and terms such as reactive oxygen species or ROS. 37, 38, but Helmut has himself cautioned against such broad and undefined usage. 4. Perhaps the greatest value of Helmut's oxidative stress concept is that it had immediate currency for those who were not familiar with biological oxidation/reduction reactions. In an effect similar to the way that the discovery of superoxide dismutase by Joe McCord and Erwin Friedrich, 44, 45, convinced hardcore biochemists that free radicals had real roles to play in living organisms, Helmut's oxidative stress terminology spoke to a wide miscellany of biologists and physicians. I am convinced that this accessibility engendered by Helmut's oxidative stress terminology was a significant factor in gaining respectability for the fledgling free radical field of storks and the West German birth rate. In 1988, Helmut demonstrated his keen understanding of the real-life significance of statistical correlations, along with a wonderfully wry and dry sense of humor. In a letter to the editor of Nature magazine, entitled A New Parameter for Sex Education, 46, Helmut suggested an explanation, in the form of a graph, for the falling birth rate in what was then the state of West Germany, an explanation he suggested, that every child knows makes sense. All Helmut presented in his letter was a simple graph showing the declining number of newborn, human, babies in West Germany between 1965 and 1980, and the declining population of breeding pairs of storks. Since the slopes of the two declining lines exhibited an almost perfect correlation, the unstated conclusion, that storks actually do deliver babies unless there are too few birds to cope, would be obvious even to children. This is a lesson I never fail to introduce to graduate students and postdoctoral fellows in my laboratory, and I highly recommend it to everyone. The oxygen paradox, oxidative stress and aging. The influence of Helmut's work on carotenoids in protecting skin against UV and singlet oxygen photodamage, e.g. 18 to 23, has been enormous. Essentially, every cosmetic company in the world has used the results of Helmut's research as the basis for producing products that prevent skin aging. One imagines that Helmut has had to deny ever making such claims himself on many occasions. Nevertheless, it does seem clear that repeated photooxidative insults to the skin impair its long-term health and vitality, and Helmut's work has shown that carotenoids can mitigate against such injuries. 18-23. Helmut has also made significant forays into the major age-related diseases of our time, including the fields of dementia, including Alzheimer's disease, 47-49, stroke, 50, and heart failure, 51. Perhaps only the fact that he remains so young at heart has kept Helmut from spending more of his time studying gerontology, although one notices that he has placed a strategic foot in the door by starting to unravel the secrets of healthy centenarians. 52. Conclusions. It is both a pleasure and a privilege to be invited to contribute to this homage to my friend, Helmut Seiss. Although my mini-review of his career hardly does justice to the broad array of major contributions he has made to science, it hopefully is sufficient to at least convince the reader that a giant is about to retire from our midst. Knowing Helmut, however, this retirement is likely just a smokescreen, or a red herring, to lure us all into complacency. It won't work, Helmut. We still expect many more great things from you in the years to come. References. 1. Seiss H. Oxidative Stress, Introductory Remarks. In, Seiss H., Editor. Oxidative Stress. Academic Press, London, 1985. Pages 1-8. Google Scholar. 2. Seiss H., Biochemistry of Oxidative Stress. Andrew Chemint et England, 1986, 25:1058-1071. Google Scholar. 3. Seiss H., Role of Metabolic H2O2 Generation, Redox Signaling and Oxidative Stress. The Journal of Biological Chemistry. 2014, 289:87-35-8741. PMC-free article, PubMed, Google Scholar. 4. Seiss H. Oxidative Stress, A Concept in Redox Biology and Medicine. Redox Biology. 2015, 4C, 180-183. PMC-free article, PubMed, Google Scholar. 5. Davies KJA, Ursini F. The Oxygen Paradox. Clup University Press, Padova, Italy, 1995. Pages 1-811. Google Scholar. 6. Seiss H., Gerstenecker C., Menzel H., Flow L. Oxidation in the NADP system and release of GSSG from hemoglobin-free perfused rat liver during peroxidatic oxidation of glutathione by hydroperoxides. FEBS Letters. 1972, 27:171-175. PubMed, Google Scholar. 7. Seiss H., Gerstenecker C., Summer K.H., Menzel H., Flow L. Glutathione-dependent hydroperoxide metabolism and associated metabolic transitions in hemoglobin-free perfused rat liver. In, Flow L., Benor H.C., Seiss H., Waller H.D., Wendell A., Editors. Glutathione. George Theme Publishers, Stuttgart, 1974. Pages 261-276. Google Scholar. 8. Flow L., Gunzler W.A. Glutathione peroxidase. In, Flow L., Benor H.C., Seiss H., Waller H.D., Wendell A., Editors. Glutathione. George Theme Publishers, Stuttgart, 1974. Pages 132-145. Google Scholar. 9. Chance B., Seiss H., Bovary A. Hydroperoxide metabolism in mammalian organs. Physiological Reviews. 1979, 59:527-605. PubMed, Google Scholar. 10. Demazy M., Sringarpura R., Davies KJA. Glutathiolation of the proteasome is enhanced by proteolytic inhibitors. Arch Biochem Biophys. 2001, 389:254, 263. PubMed, Google Scholar. 11. Ardyl Ge, Cis H. The Biochemistry of Selenium and the Glutathione System. Environtoxicol Pharmacol. 2001, 10:153, 158. PubMed, Google Scholar. 12. Mujesh G., Dumont W.W., Cis H., Chemistry of Biologically Important Synthetic Organoselenium Compounds. ChemRev 2001, 101:2125, 217. PubMed, Google Scholar. 13. Steinbrenner H., Speckman B., Pinto A., Cis H. High selenium intake and increased diabetes risk. Experimental evidence for interplay between selenium and carbohydrate metabolism. J. Clin Biochem Neutre. 2011, 48:40, 45. PMC-free article, PubMed, Google Scholar. 14. Ardyl Ge, Mostert V, Ibrahim H, Berviba K, Abel J, Cis H, Protection by selenoprotein P in human plasma against peroxynitrite-mediated oxidation and nitration. J. Biochemistry 1998, 379:1201, 1205. PubMed, Google Scholar. 15. Berviba K, Rausen I, Sheriff VS, Cis H, Attenuation of oxidation and nitration reactions of peroxynitrite by selenomethionine, selenocysteine and epsilon. Biochem J, 1996, 319:13, 15. PMC-free article, PubMed, Google Scholar. 16. Muller A, Cadenas E, Graf P, Cis H. A novel biologically active selenoorganic compound, I-glutathione peroxidase-like activity in vitro and antioxidant capacity of PZ51, epsilon, Biochem Pharmacol. 1984, 33:3235, 3239. PubMed, Google Scholar. 17. Steinbrenner H, Bilgic E, Alalai L, Cis H, Brenisen P. Selenoprotein P protects endothelial cells from oxidative damage by stimulation of glutathione peroxidase expression and activity. Free Radique Res 2006, 40:936, 943. PubMed, Google Scholar. 18. Cis H. Carotenoids and tocopherols as antioxidants and singlet oxygen quenchers. J. Nitrosibitaminol, Tokyo, 1992:27, 33. Spec No, PubMed, Google Scholar. 19. Cis H, Stahl W, vitamins E and C, beta-carotene, and other carotenoids as antioxidants. AMJ Clin Neutre. 1995, 62:1315 S, 1321 S. PubMed, Google Scholar. 20. Ost O, Stahl W, Cis H, Trinea H, Heinrich U. Supplementation with tomato-based products increases lycopene, phytofluene, and phytoene levels in human serum and protects against UV light-induced erythema. In J. Vitam Neutre Res 2005, 75:54, 60. PubMed, Google Scholar. 21. D. Masio P, Kaiser S, Cis H. Lycopene as the most efficient biological carotenoid singlet oxygen quencher. Arch Biochem Biophys. 1989, 274:532, 538. PubMed, Google Scholar. 22. Heinrich U, Gartner C, Wiebusch M, Eichler O, Cis H, Trinea H, Stahl W. Supplementation with beta-carotene or a similar amount of mixed carotenoids protects humans from UV-induced erythema. J. Neutre. 2003, 133:98, 101. PubMed, Google Scholar. 23. Heinrich U, Newcomb K, Trinea H, Cis H, Stahl W. Long-term ingestion of high flavanol cocoa provides photoprotection against UV-induced erythema and improves skin condition in women. J. Notter. 2006, 136:1565, 1569. PubMed, Google Scholar. 24, High C, Djam A, Kleinbongard P, Skew T, Cease H, Kelm M, Vascular Effects of Cocoa Rich in Flavin 3 ols. JAMA. 2003, 290:1030, 1031. PubMed, Google Scholar. 25, Schroeder H, High C, Bowser J, Kleinbongard P, Keen CL, Hollenberg and K, Cease H, Quickure eBay C, Schmitz HH, Kelm M, Epicatechin Mediates Beneficial Effects of Flavanol-Rich Cocoa on Vascular Function in Humans. Proc. Nat. Academic Sci. USA 2006, 103:1024, 1029. PMC Free Article, PubMed, Google Scholar. 26, Cease H, Polyphenol and Health, Update and Perspectives. Arch Biochem Biophys. 2010, 501:2, 5. PubMed, Google Scholar. 27, Stahl W, Cease H. Lycopene, a biologically important carotenoid for humans? Arch Biochem Biophys. 1996, 336:1, 9. PubMed, Google Scholar. 28, Stefan Y, Gruber C, Skew T, Cease H. Monomethylated flavanols and other flavonoids as inhibitors of endothelial NADPH oxidase. Arch Biochem Biophys. 2008, 469:209, 219. PubMed, Google Scholar. 29, Klotz LO, Cease H, Reversible Conversion of Nitroxyl Anion to Nitric Oxide. Methods Enzymal. 2002, 349:101, 106. PubMed, Google Scholar. 30, Korth HG, Ingold KU, Sustman R, DeGroot H, Cease H, Tetramethylorthoquinodimethane, first member of a family of custom-tailored callitropic spin traps for nitric oxide. Anjou Chem and Ed England, 1992, 31:891, 893. Google Scholar. 31, Murphy Me, Cease H, Reversible Conversion of Nitroxyl Anion to Nitric Oxide by Superoxide Dismutase. Proc. Nat. Academic Sci. USA 1991, 88:10860, 10864. PMC Free Article, PubMed, Google Scholar. 32, Stefan Y, Skew T, Cease H, Epicatechin elevates nitric oxide in endothelial cells via inhibition of NADPH oxidase. Biochem Biophys Residential Commune. 2007, 359:828, 833. PubMed, Google Scholar. 33, Sushjek CV, Skew T, Cease H, Cronky KD. Nitrite, a naturally occurring precursor of nitric oxide that acts like a prodrug. J. Biochemistry 2006, 387:499, 506. PubMed, Google Scholar. 34, Cease H, Shara VS, Klotz LO, Brviba K. Glutathione peroxidase protects against peroxynitrite-mediated oxidations. A new function for selenoproteins as peroxynitrite reductase. J. Biochemistry 1997, 272:27812, 27817. PubMed, Google Scholar. 35, Brviba K, Klotz LO, Cease H, Defenses against peroxynitrite. Methods Enzymal. 1999, 301:301, 311. PubMed, Google Scholar. 36, Masimoto H, Kissner R, Copanol WH, Cease H, Kinetic Study of the Reaction of Epsilon with Peroxynitrite. FEBS Let. 1996, 398:179, 182. PubMed, Google Scholar. 37, Kulyan Rahman B, Darli Esmer V, Davies KJA, Dennery PA, Foreman HJ, Grisham MB, Mann GE, Moore K, Roberts LJ, 2, Iskaropoulos H, measuring reactive oxygen and nitrogen species with fluorescent probes, challenges and limitations. Free Radique Biol Med 2012, 52:1, 6. PMC Free Article, PubMed, Google Scholar. 38, Foreman H.J., Augusto O., Brigelius Flo R., Dennery P.A., Kulyan Rahman B., Iskaropoulos H., Manji E., Roddy R., Roberts L.J., 2, Dina J., Davies K.J.A. Even free radicals should follow some rules, a suggested guide to free radical terminology and methodology. Free Radique Biol Med 2015, 78:233, 235. PubMed, Google Scholar. 39, Davies K.J.A. Oxidative Stress, The Paradox of Aerobic Life. Biochem SOC SYMP. 1995, 61:1, 31. PubMed, Google Scholar. 40, Davies K.J.A. The Broad Spectrum of Responses to Oxidants in Proliferating Cells, A New Paradigm for Oxidative Stress. IUBMB Life. 1999, 48:41, 47. PubMed, Google Scholar. 41, Davies K.J.A. An Overview of Oxidative Stress. IUBMB Life. 2000, 50:1, 4. PubMed, Google Scholar. 42, Davies K.J.A. Oxidative Stress, Antioxidant Defenses, and Damage Removal, Repair, and Replacement Systems. IUBMB Life. 2000, 50:279, 289. PubMed, Google Scholar. 43, Davies K.J.A. Degradation of Oxidized Proteins by the 20S Proteasome. Biochemie. 2001, 83:301, 310. PubMed, Google Scholar. 44, McCord J.M., Fritovich I. Superoxide dismutase, an enzymic function for erythrocuprion, hemacuprion, Journal of Biological Chemistry. 1969, 244:6049, 6055. PubMed, Google Scholar. 45, Fritovich I. The Trail to Superoxide Dismutase. Protein Science. 1998, 7:2688, 2690. PMC Free Article, PubMed, Google Scholar. 46, Cis-H. A New Parameter for Sex Education. Nature. 1988, 332:495. Google Scholar. 47, Lee L., Willits R.S., Polidori M.C., Stahl W., Nels G., Cis-H., Griffiths H.R. Oxidative LDL modification is increased in vascular dementia and is inversely associated with cognitive performance. Free Radique Res 2010, 44:241, 248. PubMed, Google Scholar. 48, Polidori M.C., Mattioli P., Aldred S., Cecchetti R., Stahl W., Griffiths H., Senin U., Cis-H., McCauchy P. Plasma antioxidant status, immunoglobulin G oxidation and lipid peroxidation in demented patients, relevance to Alzheimer's disease and vascular dementia. Dement geriatricone disorder. 2004, 18:265, 270. PubMed, Google Scholar. 49, Polidori M.C., Pertico D., Mangialash F., Mariani E., Ost O., Enlisik T., Meng N., Peinke L., Stahl W., Cis-H., McCauchy P., Nels G. High fruit and vegetable intake is positively correlated with antioxidant status and cognitive performance in healthy subjects. J. Alzheimer's, Dis 2009, 17:921, 927. PubMed, Google Scholar. 50, Polidori M.C., Cherubini A., Stahl W., Senin U., Cis-H., McCauchy P. Plasma carotenoid and malondialdehyde levels in ischemic stroke patients, relationship to early outcome. Free Radique Res 2002, 36:265, 268. PubMed, Google Scholar. 51. Polidori MC, Savino K, Aluni G, Fredio M, Senin U, Cease H, Stahl W, Makachi P. Plasmolipophilic antioxidants and malondialdehyde in congestive heart failure patients, relationship to disease severity. Free Radique Biol Med 2002, 32:148, 152. PubMed, Google Scholar. 52. Polidori MC, Makachi P, Ryman A, Cherubini A, Cecchetti R, Berviba K, Stahl W, Cease H, Senin U, Plasmolipid peroxidation and vitamin C status in healthy centenarians. J. A. M. Geriater SOC 1999, 47:1038, 1039. PubMed, Google Scholar.